Hey guys, welcome back. This is Lei. In the last video, I showed you how to perform alternative splice analysis. In this video, I'm going to show you how to identify RNA editing events using RNA-seq data. Both processes are post-transcriptional mechanisms that can change protein sequences. However, they differ in mechanisms and effects. Alternative splicing changes the protein sequences by allowing combinations of exons, while RNA editing directly changes RNA sequences. For example, it can change C to U or A to I in an RNA sequence directly. And depending where it occurs, RNA editing can either change protein sequences when it occurs in coding sequence, or regulates gene expression when it's found in non-coding sequence. Our job in this tutorial is to detect such events. The tool we're going to use is the Linux command line tool called REDItools2. The tool accepts bind files as its input and output an RNA editing events table. And hopefully you still remember what bind files are. They are alignment files when you align FASTQ files to a reference genome. The whole process can be done in one step. But before that, let's set up our environment. Here, we create a new Conda environment called env REDItools2, and it requires a very old Python version. And we activate this environment and install all essential dependencies like this. And after that, we directly download the tool from GitHub using the git clone command. And once it's downloaded, we get into the folder and install the tool using pip install. And once it's installed, we can verify the installation using this command. And next, we create our output directory using makedir. And the bind file we use here is free from our previous tutorial. And once everything is prepared, we can start setting up the parameters. Here, the main function is called REDItools.py. -s, this option means strict condition. -f is input file, it's our bind file. -r is the genome, a reference genome. Hopefully you still remember where to download it. -o is the output file, it's our events file. Once it's set, you can start executing the command. And after a couple minutes, you'll get the result. The resulted table has this format. The first column is chromosome, second position of the events. Third is the nucleotide of the reference genome. And strand, this is a quality score, another quality score, and this is important. This shows how many reads are mapped to each nucleotide. And this column represents the alternative substitutes. It's our the change, the editing size. Okay, the frequency is a proportion of the reads mapped to the substitute nucleotide against all the reads mapped to all four nucleotide. Let's take this one as an example. Here is the T to C conversion. So there are 2 reads mapped to C and 7 reads mapped to T. T is a reference nucleotide. Okay, so our frequency would be 2 divided by 2 plus 7. That's 22%. This is how we calculate the frequency. If your goal is to identify editing events, you can stop right here and you already have everything. But if you want to compare between different groups and you have multiple samples, and we can do a differential editing analysis, just as we did the analysis for genes. So the analysis can be done in R, and we need the packages like we had in our D analysis. We need the data table, limma, and ADAR they are the package for differential analysis. So in my case, I have 4 samples. So I have 4 editing tables. So the code looks complicated, but for now, let's just focus on the meaning of each block of code. So let's first see the first block. So here I just read in all 4 editing tables in R and store into another variable called ready underscore dt. And this is a list. And the list contains 4 tables here, 4 tables. Each table has this format. And the event ID, basically we just pasted the site coordinate and the type of site here. We paste region, position, and this column as its event ID. And we only took the frequency column because we want to perform differential analysis on the frequencies here. So basically, we read in all 4 tables and remake event ID, and we took all the frequency columns here. And next, we merge all 4 tables into one, like this. So this table has all the editing sites and the frequencies in each sample. And before we do our DE analysis, we have to normalize the proportion data because limma only accepts the normally distributed data. But obviously, our proportion data is not normally distributed. So here we use a normalization method called Arcsine square root method. So now let's normalize all the proportion values using this method. And don't forget, we need to prepare a sample information table like we did in the differential gene expression analysis. So here is the sample information table called meta look like. And now we combine the editing site table and the sample table into a DGE list object. You remember the object for differential gene expression, so it's easier to access both tables. Once combined, we can do the differential analysis exactly the same way as we do for the differential expression analysis. So the resulted differential editing site table is like this. Looks familiar? It's exactly the same format as our differential gene expression analysis. So here we only need to look at two columns, log FC and adjust p-value. So log FC is not log fold change anymore. It's just difference between the frequencies between the two groups. The adjust p-value, here we don't have any significant results. But here we have a problem. These are not genes. They are coordinates of the editing sites. But how do we connect them with genes? So our next step is to annotate these coordinates. So we need to reformat our differential editing sites by making a bat file, a bat format like this. So if you're sharp, you may have already noticed that this bat file is actually the breakdown of our coordinates in the first column of our differential editing table. So it's just the first column, the chromosome, second is the position, third is the same position because it's individual site. So we make this format, and next we fetch our genome annotation file, the GTF file. This is from the mouse. We convert the same GTF file into a bat format too, so that we can overlap them. Once converted, we can annotate our editing sites differential editing sites using the REDItools intersect command. So -A is our differential editing site bed file. -B is the annotating bed file we just made. And we store the results into a new bed file in this format. And now you can see that we have chromosome, our position, and the location, type, and the genes associated with this location. And now you have everything. You have differential editing sites and genes associated with it. So you can filter it. And these bed files are actually tab delimited tables. They can be opened in Microsoft Excel. And that's all we have for this tutorial. And now you can go to my website and copy out the code. It doesn't matter if you don't understand it. Just go ahead and try it. And you can use ChatGPT to explain or interpret the code you don't understand. We learn by doing it, not by reading it. In the next video, I'm going to show you how to do isoform level analysis.